Welcome to session 15D, Origin of Life on Earth and Beyond. This is one of the most exciting areas of science for which geochemistry is absolutely central, and we hope that over the next hour you'll get a chance to see how that works and ask questions to help illuminate all that. I'm Ariel Anbar. I'm co-hosting the session along with Sarah Russell, and we want to, at the outset, give some thanks to Adina Paitan, who helped organize this session. Hopefully you had a chance to watch the great lectures from our panelists, Natalie Batalla, David Deemer, Tori Holler, and Hillary Hartnett. And we're sure you have many questions. And so what we invite you to do is to write those questions in the question section or the chat of the GoToMeeting system. Also, we'll be willing to call on you if you want to raise your hand later on. What we're going to do, though, first is give our speakers a chance to very briefly recap their talks. We're going to ask some questions. Sarah and I will ask some questions, and that will consume about the first half of the hour, and then we'll shift to questions from all of you. We organized those 4 talks in a loose progression as follows. First, you heard from Natalie, who gave an overview of the state of the science of exoplanet hunting. Then Dave gave a presentation giving a look at a cutting edge and somewhat controversial idea about where and how life began. Tori gave a perspective on the factors that we need to consider when looking for life, especially beyond just water, particularly in our solar system. And then Hillary gave an overview of the sort of things that we have to think about as geoscientists when looking for life on a planetary scale. Sarah, I'm going to turn it over to you to go a little deeper with our panelists.
Okay, thanks, Ariel. So as Ariel said, we hope you've had a chance to look at these fantastic talks. But if you haven't, firstly, it's not too late. You can watch them at any time. But just to get everyone up to speed, we're going to ask each of the panelists to give a 1-minute lightning summary of what they said in their talk, followed by a couple of focus questions just for their talk. So I'm going to start off with Natalie, please.
Thanks, Sarah. And thanks for the invitation. I wish that we could all be together in Hawaii, but I'm appreciative of the efforts to enable this virtual forum for us to talk. So I really appreciate it, and I'm glad to be here. My talk was on exoplanets. These are planets orbiting stars beyond the solar system, other stars in the galaxy. And it was just 10 years ago, we only knew of about 200 planets orbiting other stars, and about 90% of them were larger than Neptune, so really not the kinds of planets that we would expect to find life on. Now, fast forward, we've had NASA's Kepler mission, which gave us about 4,000 new planet discoveries, and now about 90% of the planets known to humans are smaller than Neptune, including a lot of Earth-sized planets and even what we call Goldilocks planets that have orbits similar to Earth in that they receive about the same amount of energy that Earth receives from its sun. So from that cornucopia of new planet discoveries, we can do demographic studies, and we've learned that planets, small planets in particular, are very common. On average, every sun-like star has at least one. But the other thing we've learned is that the diversity of planets in our galaxy far exceeds the diversity of planets in our solar system. And there are hints that that diversity extends down to the terrestrial planets like Earth, which of course has strong implications for life, or for the propensity of life in the solar system. But right now we only have the bulk properties that we measure of the exoplanets, things like the orbital period or the radius or the total mass. And there's a limit to what we can learn about the diversity of planets from just those bulk properties. But luckily, in the future, we're going to see a new epoch of exoplanet exploration begin, and that's the epoch of atmospheric characterization. The James Webb Space Telescope, which is the successor to Hubble, will be launching next year, and it will have the ability to detect chemical species in the atmospheres of other planets. And through those measurements, we will be able to better understand the diversity of all planets, including some of the small planets. Not all terrestrial-sized planets that are orbiting in the Goldilocks zone are necessarily habitable environments. And as we search for water to find the possibility of life, maybe too much water can be too much of a good thing. So those are the kinds of questions that we want to answer, with the ultimate goal one day of being able to detect not just a planet, not just a planet in the Goldilocks zone, or even a potentially habitable environment, but a truly living world. Thanks.
Brilliant. Thank you so much, Natalie. That was it was an amazing talk, and it sounds like the James Webb Space Telescope is going to be a real game changer for your field. So could you say a little bit more about why it's so important, and how it's going to look at the composition of atmospheres, planetary atmospheres?
Absolutely. The James Webb Space Telescope is a 6.5 meter aperture telescope compared to Hubble, which is about 2 meters. So we have significantly more collecting area. But Webb will also operate in the infrared, and it will cover from the red part of the visible spectrum all the way out to 10 microns. And in that very broad wavelength regime, we can observe the molecular features of water vapor in the atmosphere, but other greenhouse gases like carbon dioxide and methane. So we will have direct measurements of important diagnostics like the carbon to oxygen ratio, or the total metallicity, which is the mass fraction of all elements heavier than hydrogen. And those are very important diagnostics for understanding the physical processes that drive planetary diversity.
Great, thank you. And so as geochemists and astrobiologists, we're particularly interested in the terrestrial planet range, and you said that you thought there might be quite a wide diversity of there. Can you say a bit more about that?
Yeah, I said that we measure bulk properties right now, like mass and radius. If you have both mass and radius, you can get the average density. And then you can run a theoretical model of a planet with a certain recipe, you know, how much rock, how much silicates versus iron versus magnesium. Add in a little bit of ice. If the ice is perhaps a planet formed further out in a region or an environment where there was the availability of more ices. So we run those models and we can try to match the mass and the radius for a given recipe. And it's that kind of data that is giving us some hint that even the small planets are more diverse than we thought.
Brilliant, thank you so much. Okay, so shall we move on to Dave? Could you tell us a bit about your talk?
Okay, as Natalie was talking, we heard the word water, you know, look for the water. So that's really been part of our definition of habitability. And since Earth was a habitable planet, I mean, here we are, it had to have liquid water. Now, most of Earth's water today is in a global ocean. A little bit, though, is fresh water. So there actually are two kinds of water that we have to keep in mind. Since so much of the water is in the ocean, we've always assumed that life began in the ocean. Hydrothermal vents came along as a place where a lot of interesting chemistry can happen. So the interest shifted to the hydrothermal vents as a possible site. I'm a biophysicist, and I am interested in more than just the chemistry of the origin of life. I want to know how those chemicals could become organized into systems of molecules that have the properties of life. Those systems have two main properties. They are polymers. The polymers are catalysts, or they carry information, and they're in a closed space, the cell membrane. So I'm interested in the origin of life, and we are able to do biophysical experiments about how polymers can be synthesized and how they can be encapsulated. When I began to work on this 10, 15 years ago, we did try seawater. We thought, sure, let's see whether seawater can help these things along. But there's some problems with seawater having to do with the biophysics and the thermodynamics of what we want to see happen in terms of polymerization and encapsulation. So we began to work with distilled water. This is rainwater, falls on volcanic land masses, and we think that the kinds of properties of volcanic land masses operating on liquid water can help life get started. So that's what I talked about, and that's what I will be happy to answer questions about now.
Brilliant, thank you very much, Dave. And in your talk, you talk a little bit about doing actual field work to try out your experiments in a real world situation. Can you say a bit more about that?
Sure. We do work in the laboratory. We have pure water that's buffered at a certain pH. We have pure molecules we're putting in to make them react. Very controlled. That's the way science is. You really want to control as many systems that you're working on as you can. But it's a big assumption to say, okay, it worked in the lab, so it must have happened 4 billion years ago. I think it's time to start testing that assumption. So we are taking what we see in the laboratory and we're going to places like Kamchatka in Russia, like Bumpus Hill here in Northern California on Mount Lassen, and most recently we were at Hell's Gate in New Zealand running our experiments. And in my talk, toward the end of it, I showed that in fact we seem to be making polymers in natural conditions as well as in the laboratory. So far so good. We're going to keep testing that.
Brilliant, thank you. And what do you think are the implications for your work for other places in the solar system? Do you think other planets or moons might host suitable environments?
I'm going to hope somebody answers the telephone. So
not mine.
Somebody did. Okay, great. Okay, other planets. Suppose, we have a 50-50 chance of being right. Life either began in the ocean or began in fresh water. Let's say that the evidence piles up and finally a consensus is that really we do need fresh water, we do need the wet-dry cycles, we do need the kinds of energy made available under those conditions. That is going to exclude certain planetary objects like the icy moons, Enceladus and Europa, but it will include early Mars. Early Mars had volcanoes. It had shallow seas. It would have had evaporation and rain on these volcanic land masses. So Mars very definitely was a habitable planet back then. I'm not going to be surprised if a few years from now, maybe rather sooner, we do have evidence that life at least once existed on Mars and might still be there yet.
Fascinating. Great. Thank you very much, Dave. And next, we're going to move on to Tori. But just before you start, Tori, I forgot to say earlier, anyone in the audience who wants to ask a question, just fire away and we'll get through as many of them as we can. And we've already got some questions coming in, which is great. Okay, so over to you, Tori. Could you tell us a bit about your talk, please?
Yeah, thank you, Sarah, and hi, everyone. So my talk was really my take on what should come next in the exploration of our own solar system if life is your reference frame. And it's certainly not the only reference frame for our solar system, but it's growing in emphasis. And for the last 2 decades, it's really left its mark on the way that we pursued planetary exploration. In particular, we've followed the water for about 2 decades, and that search has been successful enough that we've identified multiple worlds where there's solid evidence for the presence of liquid water, either now or in the past. And so it's important to ask, with that sort of abundance of riches, what do you do next in an ultimate search for evidence of life? So in my view, it's a few things. The first is to look beyond water, as it's not the only requirement for life. Life requires energy and certain elements and a particular range of conditions, and that's begun to happen. So some of our missions have begun to look at those other boxes, but it's insufficient, I think, to simply ask, are those things satisfied? Instead, I think the next step is to ask how robustly are they satisfied? Because the presence of life in a world doesn't guarantee an ability to detect it. And so you want to focus your search, I think, on the places where the supply of life's needs is so robust that it gives you a big signal to look at. And that idea of detectability is something that both Hillary and I focus on in our work. I think because of the different focus, exoplanets and solar system, the way we pursue it is a little bit different, but the idea is very much the same. And then I think finally, the thing that we can do, everything that we conceive about the idea of habitability or even detectability is based on a conception of life as we know it, and that means extant life as we know it, with a particular set of capabilities and a particular set of requirements. And I think that if we relax that life as we know it requirement a little bit and start thinking about places in reference maybe to the origin of life, how might the requirements have differed? How might the capabilities have differed? It may give us a different window into what's possible. And I think that this speaks a little bit to what Dave just mentioned. If you can convince yourself that you understand something about the conditions that foster the origin of life, it informs your sense of possibility and maybe then your sense of where to look.
Great, thank you, Tori. So how far do you think we can push this idea of life as we know it? How much can we expand it to include extra things that are different to our own life?
Yeah, I think so. I think first of all, there's quite a long way that we can go before we have to relax that assumption, because we haven't nearly exhausted the ability to think about environments in terms of their capacity to satisfy our kind of life's needs. But what's worthwhile to say about life as we know it, it doesn't just mean a certain set of requirements, which may differ for different kinds of biochemistry. So perhaps we can look in a broader range of places, at a broader range of elemental requirements, or solvent requirements, whatever. But I think it also means a different set of capabilities. So our modern extant life, even the simplest cells, are extraordinarily capable things, right? They catalyze reactions millions of times faster than they would happen. They compartmentalize themselves from the outside world. They can keep the inside conditions different than the outside conditions. And it gives them an ability to proliferate into a whole diversity of niches that might not be accessible to the origin of life. And so I think that trying to relax our assumptions a little bit to think about life that is not as capable as our own becomes important in the way that we think about the solar system.
Oh.
Did you lose me?
No, we got you. Oh,
Hello?
we lost. I think we lost Sarah.
Uh-oh.
Sarah, are you still with us?
Hello? We can hear her.
We can hear you, Sarah. So if you want to keep on going, even though we can't see you.
Oh, no.
No, her mic apparently is off. I'm trying to turn it on again.
Well, we could, I can move on to Hillary while we're waiting for Sarah to come back online. So Hillary, would you like to give a little lightning summary of your presentation?
Sure, I'd be happy to. And thank you so much for the opportunity to be part of this panel. It's been a lot of fun to kind of see how all these talks sort of integrate and interact and address lots of the same themes. So my talk is really the result of what happens when a biogeochemist or an oceanographer starts talking to astrophysicists and geophysicists around this question of exoplanets. And I had two main messages. The first is that elements that are really important for biology, and in my case, I'm thinking about phosphorus and nitrogen predominantly, are not always extremely well known throughout the galaxy, in part for phosphorus, because it's difficult to measure in stars. And so if we have to think about exoplanets as getting their recipe of ingredients, to take a phrase from Natalie, from their stars, there are important elements for life that we don't actually know much about in stars. And so we need more data, right? There's actually fundamental data limitation on how much phosphorus is available from stars. And if we want to then partition that phosphorus through models at this stage into how planets are formed and how plate tectonics get started and how, assuming that life evolved on that planet, we're even all the way back at step one, what's the recipe of elements that a planet can get? We don't know the answers to that yet. So we need to know more about bioessential elements in stars. The second part of my talk, I took the perspective that we have to think about planets as systems, ecosystems even, right? Life on Earth does not exist separately from the geological system that is the planet. And so if we want to think about whether a molecule like oxygen in an atmosphere is a signature of life, we really need to understand what the planet itself does that affects oxygen in the atmosphere of a planet. You can make oxygen abiotically through photolysis of water. So if you have a star with UV light and water, you can have oxygen in the atmosphere. Now, if you also have life and it's able to do photosynthesis like the algae in our ocean, you might have a lot more oxygen. And so we developed this index, we call the Detectability Index, which allows us to begin to quantify the production of oxygen by abiotic processes against the production of oxygen by biology, or what we anticipate biology could be capable of if it was like ours. And that opens up the possibility of being able to prioritize those planets where we think the potential for detectability of oxygen, meaning life, is higher than those planets where it might be that oxygen could come from something that's not biological. And we tested that against a planet that we call a water world, where it has an ocean with no continents that are exposed above the surface. And when we look at that kind of an ocean world, we realize that there's no continents exposed, and so there isn't any weathering of the land to supply the nutrients to the ocean the way it works on Earth. And so a planet that has an ocean, a whole ocean planet, could be perfectly habitable, close enough to its star to be warm, could have nutrients from the rocks, no problem, but it may not have enough of, say, a specific nutrient like phosphorus for biology to have a big enough imprint on the planet that we could detect it through its atmospheric signal. This was a hypothetical planet. We don't know for sure if that's what it's going to look like, but we're beginning to try to figure out how do we balance kind of geological processes, geochemical processes against biological processes so that we can begin to be a little bit more predictive about what biosignatures might be good on which planets.
Thanks. Sarah, would you like to take over
sorry,
again?
I dropped out. I missed the end of Tori's answer, which I'm very disappointed about, but thank you very much, Hilary, for that summary of your talk. It's great. For looking at the abundance of elements in the stars, and particularly phosphorus, can you use modeling to try and help inform you? Do you have a handle on how bioessential elements might vary during galactic evolution, for example? Is that kind of...
So that's a great question. There's a couple of parts built into that. Certainly there are astrophysicists, not me, who can model the galactic nucleosynthesis and understand what elements are formed at what stages, which elements are formed together. And so we might be able to make some predictions about what elements we expect to track with each other, right? Potentially phosphorus and silicon should have roughly the same distributions because of how they form. Other elements like rock forming elements, say iron or magnesium, might not have the same pattern with phosphorus. One of our biggest problems is that we don't measure phosphorus in stars very easily. And so there's a lot more data for elements like magnesium and iron and silicon than there is for phosphorus.
Sure, thank you. And then secondly, if I may ask a question that came up from the audience in our last session, but we didn't get a chance to ask you. It was, what if your water world has plate tectonics, and maybe how would that change things? So perhaps the mountains would keep growing until they're above the surface of the water anyway, or plate tectonics might have some other effects.
Well, one of the beauties of being a modeler is that you can decide what your planet's going to look like, right? And so we chose a planet that was as Earth-like as possible, except that it had enough water to bury its continents. We didn't presume that it did not have plate tectonics, just that it had no exposed continents. And what this really kind of highlights is that we need to be talking to our geophysics and petrology colleagues about how do planets work, right? What are the processes that cause plate tectonics and redistribute elements? Does it matter if you have the kind of subduction zone tectonics that we have on Earth? Does it work if you just have what they call stagnant lid tectonics? How much recycling of elements do you need? How does water interact with the rocks? It's actually a really complicated question about how planets evolve. And so for our model, we assumed that there probably was hydrothermal circulation, We assumed that the continents existed, but just weren't exposed to what we call subaerial weathering, where water rains down and breaks up rocks and it flows to the ocean.
Great.
Does that answer the question?
Yeah, absolutely. Yeah, thank you. Great. So I'll pass you all back over to Ariel to broaden the discussion out a little bit.
Great. So we have some broad questions, but before we do that, we have some very nice focused questions coming in from the audience. So I think we'll pivot to those. So this is a question primarily for Natalie. Will the Webb telescope be capable of resolving binary planets like our Earth-Moon system? It's a question from Peter McGoldrick.
No. So by resolving what the issue that's raising is direct imaging, or the question it's raising is one of direct imaging. Will Webb be used to actually image planets, to separate out the light of the parent star from the light of the planet and see either the thermal or reflected emission from the planet? And Webb is expected to do some of that for very large planets in very broad orbits, very, very young, hot planets as well. Not for planets like Earth. For that, you need a contrast sensitivity of like 10 billion to 1 in your detector technology, but you also need to have technology that separates out or suppresses the starlight so that you can see the faint signal coming from the planet. And those are technologies that are used, but currently don't have the sensitivity for an Earth. So we need to get to that state of technology before we can get to the technology where we could resolve an Earth and a moon, which would be even harder. Well, once we do resolve an Earth and we can catch the reflected light from it and analyze it, it will be an unresolved fuzzy dot. It won't be a resolved image. We won't be able to see the Earth or the terrestrial planet as an actual sphere. It'll just be a blurred image. So doing that for resolving an Earth and a moon is going to be very difficult. Probably that will require space-based interferometers many decades in the future.
Great, and teeing off that, I realize this is outside of most of your areas. I'm curious if anybody wants to say anything about the relevance, possible relevance of a large moon, of a binary planet, the origin of life. There's been some speculation about that over the years. Tides and such.
Well, you mean in terms of stabilizing the spin axis? Like
stabilizing climate, creating tides that
right.
can lead to wetting and drying cycles, perhaps, tying to Dave's talk.
So I would be willing to think about it this way. I'm not going to speculate about what the moon does when it's there, but if you form the moon the way we form the moon, right, relatively early, it may have an important implications for redistributing elements that may have been isolated in the core or deep in the mantle after the planet differentiated, right? If all of our phosphorus is tied up with iron in the core, right, and you can redistribute it by melting a large chunk of rock, that might be really important.
So also a question by Peter McGoldrick, this one is for Dave. What about littoral marine hydrothermal fields? He speculates that there might have been a lot of them on an early Earth and with an ocean composition different from today. So
a marine littoral hydrothermal field, I assume that means something happening in an intertidal zone. Is that the meaning of that?
That's correct. And since this question comes from Peter, who also asked about detecting binary planets, I imagine he might be thinking about tides coming in and out and hydrothermal environments, those kind of settings.
Yeah, well, let's just answer the literal one first. What that would do is to provide, which we still have today, wet dry cycles in intertidal zones. And having it as a hydrothermal field means that it's a hot region of an intertidal zone. So that would provide activation energy just by the heat, more than we would get from, say, where we are today with most intertidal zones just going through wet dry cycles. Now, what happens at the molecular level as salt water dries is that the salt gets extremely concentrated and finally literally begins to form crystals. Organic compounds begin to get trapped between the crystals and if there are divalent cations like magnesium and calcium present, those begin to react with carboxylate groups and phosphate groups. And that just brings these things out of solution into a kind of a curd, if you will. Those are really hard to get the kind of self-assembly and organization out of that situation compared to freshwater. And we have directly compared Yellowstone freshwater with seawater in terms of the ability to support encapsulation. Seawater going through wet-dry cycles just completely disrupts the possible ways that order can be imposed on these concentrated reactants. Freshwater allows these reactants to get concentrated and to begin to self-assemble into the structures associated with the origin of life.
Great, sorry, I was muted there. There's another question that this kind of goes to Dave, but Tori, you may also want to comment on this. I mean, it's a very broad question, really. It's kind of the question that to some extent is being skirted around here. How do you go from polymers to life that reproduces itself?
That's where the field is right now. Recent paper out of Jack Szostak's lab shows that a system of encapsulated polymers literally can begin to copy itself. This is pretty exciting, and it just came out a few weeks ago, as I recall, and I was very interested in seeing that. They did have to put in, however, pre-existing short polymers that had been chemically activated to help this process along. But that is an incremental step toward our goal of having a reproducing system of polymers that can undergo reproduction in a simulated prebiotic environment. But as I say, that's where the field is right now. We're hopeful that it's going to happen. We haven't demonstrated it yet.
All right, anybody else have anything they want to add around the mystery of the origin of life?
I think the only thing I could try to add, so Dave really is the expert there. The only thing I would try to add is then that when you begin to rely on your polymers for transmitting information from generation to generation, as soon as you need to replicate information, you need to have a means of applying energy into the system in a very dedicated way. And so it adds a layer of complexity, I think. It levies a requirement not just to be able to polymerize, which, as Dave said, you can do by cycles of wetting and drying, but if you want to actually reproduce a specific set of a sequence of polymers, you levy on yourself a requirement to bring energy metabolism into the system, and that really changes the complexity of it. And I think it's the point of coupling between metabolism, the capture of energy, and polymerization and information processing.
Yeah, that's a good point. Manfred Eigen came up with the error catastrophe. If there's too much noise going into the system, a replicating system just cannot keep it up. That's a tough one to get over. Irene Shen has been approaching that question with her work. So I'm happy that we can at least get polymers long enough to begin to fold into potentially active catalysts, for instance. So we are making things long enough to become ribozymes. And that's another sort of step that we still have to get through. And that is, how does a random sequence of something fold into a structure that can have catalytic ability or can reproduce itself? This is a toughie. So every time we make a little step forward, we see a vast array of problems emerging that we're just bumping into them all the time.
All right, so I'd like to steer this to some broad questions that I think all of you have something to say about, and we'll just go in order of the presentation. So starting first with Natalie. So the search for life has tended to focus on Earth-like planets and life as we know it. I think, Natalie, you really nicely showed us that as we start to discover the diversity of planets, we're starting to deviate to broaden our perspective on what Earth-like really means. But we still tend to focus very much on life as we know it. based on water, carbon, nitrogen, phosphorus, organic molecules. But if there's one thing we've learned in space exploration, it's that nature tends to surprise us. So I'd like to know if each of you, starting with Natalie, can reflect a little bit from your particular perspective on whether or not our preconceptions about life as we know it might be limiting us in how we search for life, or not.
I mean, I think the answer is most definitely it is, right? Because we are creatures of, I mean, we are products of who we are, and how we think follows suit. But you know, what I've learned is that, you know, nature is the same everywhere, and we see the building blocks of life everywhere. And the most common elements in the universe are the elements that give rise to life. I mean, I don't think it's a coincidence that life on Earth is made of carbon. Carbon has very special properties and can form double bonds and, you know, create complex chemicals or complex molecules and all of that. Water is a very special molecule as well. It's hard to find a solvent with the properties of water that is liquid at reasonable temperatures, not too cold temperatures. So I almost feel like life on Earth is this natural manifestation of the periodic table. I mean, Dave and I have had these conversations. So in that way, I wouldn't be surprised if all life in the universe is also carbon-based and utilizes water, which is hydrogen and oxygen, again, the most common things in the universe, common elements in the universe. But when I ask my biology colleagues, you know, do you expect life to encode information as DNA? That's where things get dicey. Although, you know, we observe amino acids in space. So, you know, again, it wouldn't surprise me, but I think that we are limited by our imagination. And I think that if you find planets where the conditions are not exactly the same as Earth, I think that nature will be creative and find ways around it to build complexity and along other pathways that we haven't considered. And we have to keep an open mind to that as well.
Right. Dave, do I answer that from, especially from the perspective of the organic molecules of life?
Yeah, the carbonaceous meteorites give us insight into this. And Natalie mentioned amino acids out in space. They get caught up in the dust particles that accrete during solar system formation into what finally arrive on the Earth as carbonaceous meteorites. So this is a gift for the chemists. There are just thousands of organic compounds and meteorites that have nothing to do with life until they come to the Earth. And we as living organisms say, holy cow, look at that stuff. You know, there's just a laboratory worth of organic compounds. Amino acids, there's 70 different amino acids, of which we use about 10 of them in life on Earth. There are nucleobases, adenine, guanine, some of the pyrimidines. There are sugar molecules in the meteorites, and all of this just done for free and then delivered to the Earth. And the message is, look how easy it is to make the molecules of life from these common elements that you're pointing out, Natalie, common elements. And that's really almost spooky that life can arise so easily, at least, you know, in our imagination, from things that are falling out of the sky and sort of telling us how it could have begun.
Falling out of the sky, and the things, the building blocks of planets.
Yeah.
That means that they were in our Earth at its beginning.
Tori, life as we don't know it, are we being limited in our thinking?
Yeah, I think this is a hard line to walk, because when you design and implement missions, you need concreteness, and concreteness comes from what you know and understand. And so I think at any point in our pursuit of exploration of our solar system or beyond, we need to be able to lock in a notion of what we think we can rely on as a notion of life or as a notion of possibility. And for that reason, we really do need to refer to what we have in front of us as an example. On the other hand, we want to make sure that we don't miss anything, and that's where it becomes important to relax our assumptions about life as we know it. And I think one of the challenges is, you know, for me anyway, the more I've learned about biochemistry, the more difficult it is to imagine doing anything a different way, because I understand I begin to understand the details of the system at such a level that it seems, well, how is this possible any other way? It's such an intricately constructed system. How could it be possible? And as you walk down the list of elements that life comprises itself of and ask about their specific functions, it becomes challenging to see those functions being served in the same way by other elements. On the other hand, if you take a step back and ask, what are truly the essential functions? Is everything that we do absolutely necessary? Or what really is? So a few things probably are necessary. I think that encapsulation probably is necessary. I think that polymers probably are necessary. But how you satisfy those things becomes a different story. And Natalie alluded to this, that the more specific you get in our biochemistry, the greater the probability that something else can do the same job. It's the really truly basic functions, the one that you believe to be universal, where you need to ask, right? Is our version of it, is the sort of energy that we use or the particular elements that we use the only way to do it? And I think that analysis, people have done it in bits and pieces, but a really, really in-depth, detailed analysis remains to be done.
Hillary, you want to tackle this question from the perspective of chemical elements?
So, life as we know it is the one that at least we know what the signals are, right? But life as we know it co-evolved with this planet from nearly the very earliest stages of the planet, right? We think that once the hot, melty Hadean Earth cooled down enough that there were continents, that life got a foothold pretty early. And so the concept that much of biochemistry reflects chemistry that we know the Earth can do, or molecules that we know can be formed under the conditions that were present on the early Earth, that makes sense, right? The thermodynamics and the geochemistry that the planet allow is the biochemistry that our planet evolved. So it's possible that planets that are slightly different might have slightly different geochemistries, and therefore life might not look like ours. But as Natalie said, there are some basic principles we know, right? Carbon, oxygen, hydrogen are going to be big players. It's because they are formed in great abundance. So I do think we should be thoughtful that if we begin to understand that not all planets have the same composition, and therefore not all biogeochemistries are going to be the same, we should be aware of that. I have a colleague at ASU, you know her, Sarah Walker, who comes from physics and as much philosophy, who thinks that we should be looking for other kinds of signals of life altogether, right? That we should be looking for how life changes information. And so you can see this in the distribution of organic molecules that life makes versus the distribution of organic molecules that we find in meteorites. The patterns in the kinds of molecules that are possible are different. I don't know what they mean, but maybe there's things that we should be considering as we start to think about broadening our envelope for what life looks like or what life might imprint on a system a little differently. I think the planet that it's on is going to make it.
Well, it's a nice segue to another big question here that I think all of you have something to say about. Is there a smoking gun? What should we be looking for, and how should we be going about interpreting the sort of evidence that we are looking for? What are false positives and false negatives that we need to worry about as we look for evidence of life? Natalie, do you want to take that first from the astronomy perspective?
Yeah, I mean, the answer to that question depends on where you're looking for life. There are three big pathways for finding life. One is to poke around in the solar system. Another is to look at the distant exoplanets that we're finding, the planets orbiting other stars. And the third is to do something like the SETI searches, where you're looking for signs of technology. What I'm focused on is looking at exoplanets. So these are planets that are, dozens of light years away. We're not going to go there anytime soon to collect samples or take result images. So everything we know is going to come from chemical markers from their atmosphere. That means that we're not going to be finding life hiding in niches or under a subsurface ocean or in a deep cave. This is got to be a world that has a global biosphere, whereby life, through its metabolism, is leaking elements into the atmosphere, and we will try to detect those that evidence of life. And right now, the primary biosignature is oxygen, because that's what's pumped into our own atmosphere with photosynthesis. So we've looked critically at this, and we still think oxygen is a very good candidate for a biosignature, but we also recognize that there are ways of producing a lot of oxygen abundance in an atmosphere. For example, through photolysis of water vapor, you can break apart water, hydrogen can escape, and you can leave a remnant oxygen abundance building up in your atmosphere. So in order to disentangle that, we have to understand the context of the planet, what kind of a star it's orbiting, what the photochemistry is, what its evolutionary history is. And, you know, at the end of the day, I think that we can't say much unless we have a statistical overview of many planets and understand the background against which we are trying to detect these biosignatures, these metabolic markers in the atmosphere. And in order to do that, we have to study many dozens of exoplanets that are both terrestrial sized and in the Goldilocks zone, in the hopes that the living worlds are going to stick out in some way. They're going to be markedly different in a statistical sense.
Almost a statistical approach to searching for life. That's really cool, and not the way people usually think about it. Dave, what should we be looking for? Is there a smoking gun? How easily can we be fooled?
I've got a couple of ideas. One is an old idea, and it has to do with what's called chirality. Now, chirality is a word that's very familiar to organic chemists and biochemists. It has to do with the fact that some molecules, in particular amino acids, have a handedness to them. And chirality actually comes from a Greek word, I believe, for handedness. So I have a right hand, a left hand, you can see them here, and they are chiral. There's a right hand, a left hand. If I have a left-handed glove, it won't fit onto my right hand, okay? Despite O.J. Simpson's problems with that. Okay, now then, suppose I add a thumb here. So now I have a symmetrical right hand. I got a thumb, 4 fingers, and another thumb. Suddenly, it is not chiral because I can flip it over and I can hold it up to a mirror and I will now see the same image. It does superimpose. Now, there's only one amino acid that is not chiral, and that's glycine. And glycine is the simplest amino acid, and it just doesn't have the chemical properties of chirality. All life has chosen way in the past to be homochiral. We're all looking at each other. We have L amino acids almost exclusively in the proteins that compose us. We have D sugars, another chiral molecule, sugar molecule, in the polymers that compose nucleic acids. So a very strong biosignature would, if we pulled up some ice from so deep in Mars, we get to that point with a way that we can dig down, bring up some of that ice. And if we analyze for amino acids that might happen to be there, there's 2 possibilities. One is that they are called racemic. There is an equal amount of the D and the L form. And that's what you usually get. If you synthesize amino acids in the laboratory, it's racemic. But life has a trick. It can pick out and synthesize only L amino acids or D sugars. So we got that signal that those amino acids have a more L than D, or possibility more D than L. We would conclude that those have passed through a metabolic process, and we would call that a very strong biosignature.
Right. Thanks, Tori. Do you want to tackle this question? What we should be looking for? What makes for a good biosignature? And
sure. So I think in the simplest terms I can conceive it, a biosignture is something that you can't explain by any other means than life. And any other means is a pretty sweeping statement, right? So if you're convinced that something can only have been formed by life, it implies that you understand the entire spectrum of what's possible in the absence of life. And to me, that means that anything you would claim to be a biosignature can't be considered outside of its environmental context. And in fact, it's really important to ensure yourself that you understand that context in enough detail to understand what's possible abiotically. And that's a challenge, right? Because we're talking about places that are difficult to observe in the level of detail that is necessary to establish that context. And I think the second challenge, and this is one that's a bit more specific to our solar system exploration and not so much to astronomy, is contamination. How do you convince yourself that what you're observing isn't just something that you brought with you? It might well be explainable only by life, but is it explainable only by life in situ or something that you brought with you? And so I think, you know, it would be very interesting to think about things that are only made by life, but also not by our kind of life, right? It crosses a threshold, but we can't recognize it in our kind of life. So for example, you know, Natalie referred earlier to the idea that information processing could possibly be carried out by a different system of polymers. If you found something that looked like that kind of system, but wasn't made of the bases that our polymers are made of, that might convince you that that's a biosignature that's not contamination. And so I think both of those things are important. The last thing I would say is that the idea of a smoking gun implies that there's one and only one thing you should be looking for. And I think that if you have the ability to do it, you should seek as many lines of evidence as you can. This has been proven over and over again, you know, people, even in exploring our own distant past, but also in materials from beyond Earth, you think you found the thing that proves it, and time passes and people work on it and you find out that that's not so. And so collecting multiple lines of evidence whenever the opportunity presents itself, and as much as possible orthogonal lines of evidence, I think is really important.
Great. Hillary, smoking guns, biosignatures?
Smoking gun. So I am at heart a chemist who thinks about these things. And one of the things to remember is that life is fancy chemistry. But one of the things that life does remarkably well, and we've used the word before, is this idea of catalysis, right? It takes reactions that are absolutely perfectly thermodynamically favorable, right? It can't make them go backwards, but it can make them go faster. And so there's a kinetic component to what life does that's different from what equilibrium chemistry or geochemistry might do. And so I think we need to also begin to think about how can we change our observing practices or enhance our searches by looking for things that have a temporal component to them, right? An example is in our own atmosphere, we have CO2. And the CO2 is increasing in the atmosphere, but there are two life-driven signals in the CO2 curve, right? It has this exponentially increasing pattern with a very stable wiggle. And that exponentially increasing pattern is the human influence, right? We'd be doing great if we could watch a planet long enough to see that it had a early Anthropocene human population. But that very stable wiggle is the seasonal pattern of photosynthesis in the northern hemisphere, because our planet has seasons, right? And so we should think about, if we find a theoretically habitable exoplanet and it's and we tend to look at things edge on so that we can do the kinds of spectroscopy that are important. What if we could figure out a way to look at one that's tilted so we could look at its seasons and see if its patterns change on a seasonal basis? To me, that would be something that was, like Tori said, another line of evidence that could be really powerful in combination with the actual composition of its atmosphere, right? Volcanoes, for instance, aren't seasonal, right? So there may be changes in the CO2, but they wouldn't have the same pattern.
Mm-hmm.
So time.
And I'm muted. So I'd like to pivot to something a bit more around science and society. This is a question that Adina actually asked in the earlier session that we weren't able to get to. The science that you all do is so closely tied to the public imagination and to science fiction, to ideas that most people, that when we were graduate students, many of us weren't actually the realm of science. How, when you go about talking to the public about your science, how do you keep the conversation moored from getting unmoored into science fiction? How do you keep things locked into science fact, or at least from veering too far? Natalie, do you want to begin with you?
I find that so easy because what we're doing is so remarkable and exciting. I mean, when I started with the Kepler mission, well, let's see, the proposal writing stage was about 20 years ago. At that time, we had a ground-based observatory up at Lick Observatory where we were trying to detect giant planets from the ground using the transit method. And we were just thinking about, you know, just the milestone of detecting the first transiting planet, a giant planet, with the thought that if we put this telescope in space, one day we could detect a planet as small as an Earth. And I just find it remarkable, the change in thinking that has happened in such a short time. We've gone from just wanting that milestone of being able to detect a planet, to all of a sudden thinking about life and detecting life and having a roadmap to do so. So reality is just as remarkable as science fiction. So I don't need to fall back or keep people engaged. I can talk about all of the amazing worlds we couldn't have imagined existed. Well, some of them we did in sci-fi. You know, we have lava worlds with oceans, not of water, but of molten rock. We have circumbinary planets. We have planets the age of the galaxy itself. We have, you know, ocean worlds, or, you know, the most common type of planet known to humanity is a type of planet we don't even have in our own solar system, these purported ocean worlds, or we don't even know what to call them. So there are so many incredible things to think about and to understand that we've learned just in the last 10 years that sci-fi is taking a back seat.
All right, Dave, you want to tackle this?
Sure. Our research is sponsored by taxpayers. Taxpayers want their money to be useful. And when I try to make this argument about the work that I do, basic research in general, and specifically what I do, I talk about spin-offs. Basic research is a fount of value that time and again has changed human life. I'll just give you two examples. One is that we're using semiconductor in just doing what we're doing right now. This is semiconductor technology, which was discovered just by chance when somebody looked at a silicon crystal and discovered that it had a special kind of electrical property. Look what that has given us in terms of the ability to interact and communicate the way we are now. In my own case, I was trying to make an artificial cell. There's an enzyme inside, and I needed to feed it with some ATP to give the enzyme some energy. And I thought to myself, well, maybe if we could punch a hole in the membrane, I could let the ATP into to feed that enzyme in the liposome. So it's just a pure basic research, trying to make a little simile of an artificial cell. And then it thought to myself, knowing how molecules can interrupt electrical current, that maybe the ATP getting in would change the electrical current going through that pore. And then I said to myself, what if it was a long string of nucleotides and a piece of DNA? Maybe it would go up and down, the electrical current would go up and down in a base-specific manner. We patented that in 1998. A company picked it up, and now that company is worth $1.5 billion. They are doing nanopore sequencing, and that I can trace that right back to a basic research idea. So I think any scientist can do that. You can talk about penicillin, antibiotics, just goes on and on. So
Dave, they're going to close us down in 30 seconds. So I'm going to turn over to Sarah to wrap us up. Thank you very much. I'm sorry we didn't get to everybody.
Yeah.
Just my favorite.
Yeah, thanks very much, Dave. And yeah, sorry, Tori and Hilary, we can't get your answer to that question because we're told we're going to be shut down at exactly past. But we can carry on discussing everything on Discord. So before we go, I just want to say thank you so much to our amazing panelists. I've learned so much in this session, more stuff from the session before. So thank you so much for engaging so well. Thank you, Ariel. Thank you, Adina. Thank you to our student helper, Raphael. And for me, it's good night. I guess it's good evening for you.
Thank
you.
Thank you.
Thanks, everyone.
Thank you.
Thank you.